I'm going to discuss mitochondrial disease with you. It's going to be a general overview, but we're going to be discussing it mainly in the context of urology. So mitochondrial disease is defined as an abnormal mitochondrial function, primarily due to alterations in the mitochondrial genome. Mitochondrial disease does not pertain to one specific disease process of the human body. Instead, it affects any organ in any person at any age. The most commonly affected organs include the brain, the heart, and the muscles, and this is because these organs require a lot more energy to function. This disease is not as rare as once believed. They think that one person in every 2,500 to 3,000 suffer from mitochondrial disease. The disease affects males slightly more than females, with Caucasians and Hispanics being the most commonly affected ethnic group. The average age of onset is between 2 weeks of age to 18 years of age, with the average age of death being just over 5 years. People who develop the disease experience different levels of severity from one another. So to some patients, mitochondrial disease might just be draining or exhausting, while to others it might prove fatal. At present, mitochondrial disease treatment consists of addressing symptoms because a cure is yet to be discovered. The pathophysiology of mitochondrial disease is focused in the cell itself. Mitochondria provide the required energy for the cell to function, so they are responsible for overseeing apoptosis as well, which is natural cell death. The number of mitochondria found in each cell is determined by how much energy that the cell requires to function. So, as discussed on the previous slide, the heart, the brain, and the lung, muscles require the most cell. So the largest number of mitochondria are those in the heart, the brain, and the muscles, and those organs expend the highest amount of energy. The energy that is created by mitochondria is known as adenosine triphosphate, or ATP, and it also is heat. The energy created through a process called oxidative phosphorylation, or OXPHOS, which uses the electron transport chain and the citric acid cycle, which is illustrated in this slide. During OXPHOS, all of the oxygen consumed by the mitochondria is eventually turned into water. However, when the oxidative phosphorylation process is not fast enough and not all of the oxygen can be transformed into water, the remaining ROS, or reactive oxygen species, or free radicals, accumulate in and damage the mitochondria. Unlike DNA in a nucleus, mitochondrial DNA is not protected, so it's open to attack and mutation. Free radicals have most damaging effects on lipids, proteins, and mitochondrial DNA, also oxidative phosphorylation enzymes. The resulting damage from the free radicals ultimately decreases the mitochondrial function, and so in order to repair this, to get back to normal function, the mitochondrial cell needs to expend a greater amount of energy, which results in additional damage. So therefore, it's kind of like a catch-22. It's not working well enough, but in order to get it working better, it needs to expend more energy, but then we have more damage. Factors that impact mitochondrial disease. Mitochondrial disease is inherited genetically. So the main route of transmission is through the mother. This is due to the fact that female zygotes contain mitochondria that are segregated into daughter cells during cell division, whereas male zygotes contain no mitochondria. If an affected woman has a baby, there's a chance that she will pass along her mitochondrial DNA or mitochondrial disease to the fetus. However, the genetic probability of this is difficult to calculate due to the fact that it is nearly impossible to determine how many affected mitochondria are in an egg. Additionally, an unaffected woman can pass along her mitochondrial disease if the mitochondrial DNA were to mutate prior to cell division and the mutated mitochondrial DNA cell were to become the fertilized egg. And this would be considered a sporadic inheritance. Since it is a genetic disease, there are no environmental or lifestyle factors that attribute to developing or passing along the disease, but I can imagine that carcinogenic lifestyle such as smoking, drinking, eating unhealthy foods will maybe attribute to genetic mutation of the mitochondrial DNA. So mitochondrial DNA affects various organs throughout the body. Mutations in the mitochondrial DNA are responsible for a wide variety of diseases that range in complexity. So, for instance, mutations in the mitochondrial DNA may be responsible for disease processes with few symptoms, such as deafness, to much more complex and complicated disease processes with multiple symptoms, such as congestive heart failure. The mitochondrial encephalopathy with lactic acidosis, or MELAS, affects the neurologic system through delayed oxygen metabolism by the mutated mitochondrial DNA. This can result in long-lasting neurologic defects, including dementia, hearing loss, neuropathy, and cognitive impairments, including language, perception, and memory function. MELAS primarily manifests itself as lactic acidosis, seizures, and stroke-like episodes. Additional temporary deficits such as migraine may develop in a person with MELAS where there is an increased metabolic demand. It is thought that migraine attacks in a person with MELAS is caused by elevated neuronal excitability due to the impaired oxidative metabolism. As stated earlier, cardiac function demands a higher amount of energy, resulting in more mitochondria per cell. Additionally, this high energy demand of the heart easily lends itself to produce clinical manifestations of myocardial disease. Specifically for this clinic, mitochondrial disease and renal function is important. There are three commonly reported kidney diseases in people with MELAS. They are Tony, Debray-Fanconi, nephrotic range proteinuria, and focal segmented glomerular sclerosis, or FSGS. It is believed that TDF syndrome is caused by a breakdown in the ATP requiring potassium pumps, which are responsible for creating an electrical gradient necessary to reabsorb essential electrolytes. Additionally, it has been suggested that dysfunctional mitochondria may be responsible for individual monocyte necrosis, which ultimately changes normal renal hemodynamics. The kidneys are requiring a large amount of energy to function properly, which put them at risk for damage from mitochondrial disease. So in patients, for this clinic specifically, this will have an overwhelming effect on their kidneys, their bladder, their electrolyte balance, their urine output, their liver, so on and so forth. So I think in people who are affected with more severe forms of mitochondrial disease, they will definitely have some renal involvement and will need some help. To conclude, mitochondrial disease is attributed to many diseases with differing clinical symptoms. All of these diseases combined make mitochondrial disease the most common of metabolic diseases. This disease process all begins with the mutation of the mitochondrial DNA, which leads to a defective OXPHOS process. The process in turn decreases the cell's ability to create an appropriate amount of energy while creating harmful free radicals, and results in the specific disease process of the affected body system. As illustrated by our discussion of MELAS, a mitochondrial disease can affect many systems of the body, complicating the disease process and adding to the morbidity and mortality of the patient's specific ailment. Many factors play a role in the development of mitochondrial disease, with genetics being the most important risk factor. So I think it's very important for us to understand with our patient population that we are seeing them for one specific area, but many are involved. Any questions?